something that you add to the vaccine to enhance the immune response. Remember back when we were talking about our T cells and how our T cells, when they are being presented an antigen from a dendritic cell, that they need to have that co-stimulatory molecule being delivered as well? Well, that's what an adjuvant will do. It kind of serves to provide that co-stimulatory molecule as well, so that the enhanced response is greater. Any questions about the two types of vaccines? Yes?
You said that the attenuated was long-lived.
Yes.
Is the inactivated not?
Yes, it's not generally. Because the immune response is, because the cells don't replicate with the inactivated vaccine, the immune response is somewhat muted. You still respond, but it's not the same kind of development of memory cells that you would get if the cell was replicating in your body. So let's say that you might have, after being exposed to a live antigen that replicated in your body, you might have maybe 100 memory B cells for that particular, whereas being exposed to something that didn't replicate and inactivated, maybe your memory B cells now would only be 10. And so over time, those memory B cells could be lost, could be degraded, and so you have to get a booster shot to re-amplify the immune response. This is why tetanus has to be given every 10 years, because you lose that immunity over time. Those B cells that were generated, they eventually degrade. And there may still be a few there, but a booster shot just re-amplifies the immune response. So let's talk about immunological testing here. So we're talking about diagnostic tests, and what we're really talking about is determining whether or not you've been exposed. That's what immunological testing is all about. And if you remember, the specificity of the antibody-antigen complex allows us then to test for exposure. All we need to do is know, one, the antibody. So if we suspect that you've been exposed and you are producing the antibody, we can then expose you to a sample of your blood or something to the antigen. And if the antibody is there, it'll bind to the antigen. We can do the other as well. If we think that you have the microbe in your body and we want to test for it, if we know what the antibody is that will bind to that, we again can take a sample and expose that microbe to the antibody, and if the antibody binds, then we know you are infected with that microbe. So that antigen-antibody specificity is what allows for us to do this immunological testing to determine if you've been exposed. If you are not producing antibodies to a particular antigen, you are seronegative. Seronegative, not zero, and I might say zero and I don't mean it. Sero, try to enunciate. But if you do get exposed to an antigen, within 10 to 14 days, you will then start to produce the antibody, and you will be described as being seropositive. This is a seroconversion. And in which case, I don't need a T in there, in which case you have gone from not producing the antibody to producing the antibody. what we say here then is that your antibody titer has increased. This is the concentration of antibodies that you have circulating. So how do we go about obtaining antibodies? Starts with an animal. A lot of times we're talking about mice. It doesn't have to be a mouse. It could be a rabbit. Rabbits can be used. Horses used to be used as well, not so much now. So the animal is immunized with the agent. So now it starts to produce the antibody. And what we're going to, I'm sorry?
With the infectious agent?
Yes. With the one that we want antibodies from. Okay, so now we are going to harvest the animal's serum. And the serum is the fluid part minus the blood clotting factors. So it's minus the formed elements in the blood clotting factors. That would be the red blood cells, the white blood cells, the platelets, and then any of the proteins that are responsible for blood clotting. Those are removed. And what you're left with is what we would call a polyclonal situation in which you have multiple naive T cells, or B cells, excuse me. Why do you have multiple naive B cells? Well, go back to our agent that we introduced to the animal. Remember now that there are likely lots of different proteins on the surface of this microbe that can be recognized by the B cells. Okay, so each one of these can produce a population of naive B cells. So being exposed to a bacterium could cause the production of many antibodies, many different types of antibodies, because you have several different antigens on that agent there. The problem with polyclonal antibodies is that if this is used as a treatment, and it can be used as a treatment, the risk you run is that sometimes some of the naive B cells that might be generated from exposure to this might be similar to antigens of your own body. That could be a concern. Or they might be similar to antigens of other bacteria cells, which would then produce a false positive. And so you really wouldn't know necessarily if the result you get from exposure to a polyclonal antibody was due to the particular microbe you're testing for or one closely related. So polyclonal antibodies, while useful, have some limited scope to them. If you use the whole bacterium, you'd probably get lots of multiple naive B cells. If you use the purified toxin, then you're going to get far fewer, maybe just one or two different naive B cells, depending on the epitope that the toxin might have. Which brings us to monoclonal antibodies. monoclonal antibody happens when only one epitope is recognized. You can see the usefulness of this then. Okay, this goes back to our immunological testing. If you can create an antibody, a single antibody that will recognize a single epitope, only one population of naive B cells, then you can test whether or not an individual has been exposed to that agent that would cause the production of that antibody. The way that we go about producing these monoclonal antibodies, it's fairly cumbersome and it's expensive, but it requires isolating B cells from an immunized animal. So they isolate B cells. So go back up here where we immunized with the agent and we got a polyclonal production of antibodies. So they isolate those B cells. Then they combine them with a cancer cell, a myeloma cell. It's a plasma cell that is no longer producing the antibodies it was designed to produce, but it still continues to replicate and grow. So they take these B cells and they fuse them with these cancerous plasma cells to produce what is called a hybridoma. And so from an exposure, you might get several hybridomas produced. Now, isolate the B cells, isolate the hybridomas, I should say, from these fusions here. Excess plasma cells will die out. Those that don't fuse will die out in this population because there's an ingredient missing in the agar, and the B cells that don't fuse will also die out. So all you have are fused cells, B cell plasma cells. Now these hybridomas will, because they're cancerous, will continue to divide, but they will now produce the antibody for this B cell. Okay, so now we have these hybridomas. You may have several populations, different B cells. Now you expose those, isolate the B cells, expose them to the antigen, and those that respond, that then is the antibody that you can mass produce for your monoclonal antibody. And that monoclonal antibody then can be used for identifying organisms that you've been exposed to that might cause a specific disease. For instance, syphilis. You can expose, take a small sample of a lesion or something, expose it to the monoclonal antibody that is produced when exposed to syphilis, and if there's a reaction, you know that individual has syphilis. Any questions, any comments? Real quick, your book talks about diagnostic tests. And these are different tests. I don't know how that's shown up. These are different ways of determining whether or not an antigen or antibody is present. And some of them will also allow us to determine to what concentration. Some of these you may be familiar with. A precipitation test is one where you take the antibodies and you combine them with the antigen, and if there is a reaction, a precipitate will form. You'll get an accumulation, a cross-linking between antigen antibody, and so you'll get this large complex that forms, a precipitate that will form. And so that tells you then that that antibody was there, or that antigen, depending on which way you're going. You have to remember you can go either way. And a glutination reaction is very similar to a precipitation reaction, but in this case you're looking for a clumping of non-soluble materials. So up here we're talking about a precipitation of soluble molecules. Here we're talking about non-soluble molecules. In this case specifically we could refer to the blood testing that you might be familiar with. In that case the non-soluble materials are the red blood cells. And so when exposed to the agent, the red blood cells then clump together. So they're very similar types of testing, but one is looking at non-soluble and the other is looking at soluble. The other one I want to mention here is ELISA. ELISA stands for Enzyme Linked Immuno